Alright, today we're going to talk about this group of proteins that we call enzymes. And enzymes are what we call biological catalysts. They make things go faster. And just like Fry saying here, not sure if an enzyme or a random word that ends in A's, if you ever see a word that it doesn't end in age in biology, chances are it will be an enzyme. Okay? So let's have a go at these three quick questions. Pause the video here. Alright, so question 1, we want to know the role of this promoter and operator in the regulation of production of enzymes, is that key word, which break down lactose. Okay, so if we're talking about breaking down lactose, we're talking about the lac operon, right? And that's a stretch of DNA inside where upstream we've got the promoter, and there it is there, and then a little bit downstream we've got the operator, and then we've got the structural genes in there, all right? And then this way we're going to have that regulatory gene as well, okay? But what's going to happen in the lac operon? The promoter is the site where DNA polymerase binds. So DNA polymerase is going to bind here, and it's going to run down the lac operon and read those structural genes, right, and actually produce those enzymes to actually break down the lactose. And that's going to happen if nothing is bound to the operator, because what's the operator? It's the site where the regulatory protein binds. Okay, and why is that important? Because if we have a regulatory protein bound here to the operator, that DNA polymerase is going to be stopped there. Okay, so we're going to control that expression of those structural genes. So now we want to draw a diagram to represent the formation of a peptide bond. Let's make our lives easier for ourselves and just draw an amino acid like this. There's an AA amino acid. Oops. You obviously should have a good idea of the carboxyl group and the amine group. I'm not going to completely write down everything, but on one side of the amino acid we've got the carboxyl. Okay, and then if we have another amino acid next door, it's going to have an amine group. Oops. Amine. Okay. If we join those two amino acids together, right, what we get is that peptide bond. There it is. Peptide bond. All right. What's the name of that reaction? A condensation. Reaction or condensation? Polymerization. Okay, what does it require? It requires an input of energy, ATP, all right, and we get an output of water. Why do we get an output of water? Because the amine loses a hydrogen and the carboxyl loses an OH. Okay, so input of energy, output of water. All right, so an experiment was conducted to investigate how heat affects the rate of diffusion of small non-polar molecules into plant cells. These molecules were stained blue and were placed with plant cells in water baths at 5, 20, 40, and 80 degrees for about 5 minutes, and the cells were photographed under a microscope and the intensity of the color was measured using a colorimeter. Explain what is the independent, dependent variables and two controlled variables. The independent variable is the temperature of the water baths. Okay, the dependent variable is the intensity. Intensity of color measured. And from this, we're going to get the amount of diffusion. Okay. Two controlled variables. We can have, the type of cells, We can have, what else can we have here? The staining method. We can have the amount of water, okay, the amount of time, so 5 minutes, okay, so lots and lots of different control variable. So in the study design, we're on to the next dot point. We're going to fly through this whole dot point in one PowerPoint. It is a pretty big one though, a very important dot point, and it's all about the structure and regulation of biochemical pathways. We can't get away from chemistry completely. But what are we going to talk about? The role of enzymes as protein catalysts in biochemical pathways, the mode of action, so how they work, including what we call reversible and irreversible inhibition of their action due to competitors at the active site, and by factors including temperature, concentration, and pH. All right, and then we also have to talk about these guys, these coenzymes, ATP, NADH, and NADPH, as in their loaded and unloaded forms to actually move energy, protons, and electrons between reactions in the cell. This last dot point flows perfectly into photosynthesis and cellular respiration, which are the next two topics, all right? You can't learn them without knowing that last dot point. Well, without knowing enzymes, full stop. So who actually does need enzymes? So enzymes have a handful of roles in biological processes, such as digestion. So you've got these enzymes inside your mouth that actually break down the food that you eat. Nucleic acid replication and transcription, so there's DNA polymerase, cellular respiration, photosynthesis, two very important processes for living things, formation of pigment, and also the breakdown of pathogens. So here we have a macrophage actually breaking down a pathogen using a specific kind of enzyme called a lysase. So lots and lots of important roles enzymes have for living things. So what is an enzyme? An enzyme is a biological catalyst that are composed of one or more polypeptide chains. All right? So all enzymes are either tertiary or quaternary structured proteins, every single one. Okay? As a catalyst, enzymes actually speed up the rate of reactions. Okay, so if this is a reaction without an enzyme here on the left, on the right is a reaction with an enzyme. So reactions can happen without enzymes, but enzymes make them extremely faster. We're talking hundreds of thousands of times faster. All right. What decides the role of the enzyme is that tertiary, the 3D structure of the enzymes. Okay. Oops, backwards. All right, so when we say an enzyme is a biological catalyst, we're often talking about a little graph like this, all right? So if you have specific reactions, reactants before a reaction, all right, we actually have to provide a reaction with a little bit of energy, all right? And that little bit of energy is called the activation energy, all right? So without an enzyme, the activation energy of a reaction is really big. So if you look here, it's all the way up here, the activation energy. All right. If we provide an enzyme, we drastically reduce that activation energy to make it a lot smaller. Okay. We provide that activation energy, the reaction happens, and we get the products. Okay. That's what enzymes really do. What's the structure of an enzyme? Why is it so important? So an enzyme, every single enzyme has what we call an active site. All right? And what the active site is where the substrate or what is going to be reacting binds to. Okay? And enzymes, so we often draw them like this on the left, but really they're three-dimensional complex protein structures that look like this. All right? So for example, this specific enzyme breaks down hydrogen peroxide, all right, which is here, and hydrogen peroxide is going to fit nicely into that enzyme, okay? So we talk about the shape of an enzyme as a very important factor, all right? The enzyme's active site is complementary in shape to its specific substrate. So here, this enzyme on the right is complementary in shape to hydrogen peroxide, but also charge. Okay, so enzymes are complementary to their substrates in shape and in charge, and that's an extremely important concept for enzymes. So how do things fit for an enzyme is through what we call the induced fit model. So it's not perfect, the shape, all right? So if you were to imagine this fist can be my substrate and this can be the active site of an enzyme, right? The substrate's going to fit in there, and then the active site, the enzyme's actually going to completely hug that substrate, all right? What we call an enzyme substrate hug. No, it's not. It's what we call an enzyme substrate complex. Okay, so here we have this substrate. It's going to go into the active site. You can see its substrate. It's sort of like the active site, but not perfect. But the substrate then changes for that enzyme, and we get that reaction being catalyzed, and we get some products being formed. All right, so there's different factors that actually affect enzymes and how well they work, their activity. Okay, so the factors we're going to look at is temperature, which is an extremely important one. It actually makes up the prac that we're going to do that for your first sack. pH, which remember means something being acidic or alkaline. All right. Enzyme and substrate concentration, so how much enzyme, how much substrate we have, and then the presence of these two guys that we call inhibitors and coenzymes. All right, so temperatures, the temperature that enzymes work at, there's always an optimal temperature that enzymes can have, okay? And this just means that that's the temperature the enzyme's going to be the most efficient. It's going to work the best, all right? Below the optimal temperature, so as we go left, all right, the enzyme becomes less efficient, all right, slowly, okay? And it actually becomes what we call inactivated. Above the optimal temperature, the enzyme becomes less efficient suddenly, okay? And this is what we call the denaturation process of enzyme. All right, let's break down why, if it goes cold, it only becomes inactivated, which is something that's reversible. Yet, if it gets really hot, it becomes denatured, which is something that's irreversible. Okay? The reason why is because if it's getting really cold, all that's happening is the molecules, which were moving quite fast, start slowing down. And in that slow down process, the substrate and the enzyme aren't going to meet as often. There's inactivation. We speed that up, they're going to move fast again, and so the enzyme and the substrate will be able to meet more often, and so we're getting activation again. Denaturation, on the other hand, what happens is that 3D structure of the enzyme, because we have an enzyme being a protein, breaks down. It becomes denatured, okay? And so once broken down, that's not going to naturally just reform. And so because that's why if it gets too hot, we're not going to get the enzyme working anymore, ever. pH also has an optimal pH, okay? And this has a lot to do with the location of the action of that specific enzyme. So for example, in humans, the optimal temperature, we'll go back actually, so for humans, take a guess, optimal temperature is about 37 degrees Celsius, 36.5. Why? Human temperature is on average 36 degrees. pH, on the other hand, is going to depend very much on where it is. For example, in our mouths, we have a pH that's quite neutral, okay? And so the enzymes that are going to work there have a pH of about 6.5, 7.5. Stomach, very, very alkaline pH, so we're going to acidic pH, so we're going to have a very low optimal pH. Your intestines, on the other hand, we're going to talk about a more alkaline pH, okay? So we actually have to look at, for temperature and pH, you have to be able to read these kind of graphs, okay? So this is a very common graph for temperature, all right? So let's look at the black line first. for humans, like we said, the optimal temperature is going to be about 37, and there it is. That means, so if you look at the graph, as the temperature increases, so as we're moving right, the rate of reaction is going to change, okay? So here, for humans, the black line, the optimal temperature is going to be 37, which means that that enzyme is going to work the best then. If we're getting colder, so if we're going in that direction, we're getting that inactivation, okay? So it's a more gradual slope. On the other hand, if we get too hot, we get denaturation, okay? And the enzyme, once it's denatured, it's not going to work again. And a thermophilic, a heat-tolerant bacteria, the optimal temperature might be quite high, so about 77, okay? pH, on the other hand, we either have that optimal pH, but either side that we go, we actually get denaturation, all right? We're just going to get different optimal pHs for different enzymes. Okay, so substrate concentration is actually relatively obvious, okay? So as we increase the amount of substrate that's available, all right, the rate of a reaction is going to increase, okay? But it doesn't keep increasing. It gets to a certain point that it actually plateaus, okay? So here we're increasing the substrate, increasing the amount of stuff that's available to react, increasing, but then we get a plateau. And the reason why is simply because there's a limited amount of enzyme, all right? So what we call a limiting factor is the enzyme available. And how I want you to think of this is like this, okay? Let's say you had to go out the yard and pick up rubbish, all right? You got in trouble and you had to pick up rubbish, okay? So let's say that there isn't very much rubbish today in the yard. So as you're going around the yard, you only pick up maybe one bit of rubbish every 5 minutes, okay? Let's say on another day you had to also pick up rubbish, but the Year 7s were really, really bad and they were putting through quite a bit of rubbish out in the yard, all right? So this time you can actually pick up one bit of rubbish every minute, all right? Because there's a lot more rubbish to pick up, okay? But let's say one day both the 7, 8, and 9s were all really, really bad and they left so much rubbish out in the yard, okay? You only have 2 hands. You're not going to be able to pick up that much rubbish, okay? So you're still only picking up rubbish every minute, because there's only one of you to do that picking up the rubbish. And that works very much the same in terms of substrate concentration for enzymes. Enzyme concentration, on the other hand, all right, isn't quite the same. And the reason is because we usually only have a limited number of enzymes available compared to the substrate. The enzyme concentration is actually going to continue to increase, and the rate of the reaction is going to continue to increase as long as there's substrate available. All right. So pretty much as enzyme concentration increases, rate of reaction increases. If there's no more substrate, the reaction just simply stops. Okay. All right. Now let's talk about this thing called inhibition. Inhibition just means we're stopping something from happening, okay? Because enzymes rely on their structure to function correctly, any alteration to the structure of the active site is going to interfere with the interaction between the enzyme and the substrate, so the two of them actually meeting together, okay? We've got 2 different kinds of inhibition. The first kind is competitive inhibition, and this is actually quite simple. What happens in competitive inhibition is we have an inhibitor which is going to fit straight into the active site. Fits into the active site, the substrate can't get in, okay? This is what we call reversible inhibition. The reason it's reversible is because if we actually get enough substrate to whack this inhibitor, eventually it manages to knock out the inhibitor and we get the reaction happening again. Non-competitive inhibition, on the other hand, is what we call irreversible. Okay, and the reason why it's irreversible is because in a non-competitive inhibitor, it's going to bind to the enzyme somewhere else, somewhere that's not the active site. This means that the active site undergoes what we call a a conformational change. Right? Conformational change means that the substrate won't fit anymore into that active site. If the substrate can't fit, it can't undergo the reaction. It's not reversible though, because now there's nothing actually trying to whack this inhibitor out. Right? It's just stuck in there. Okay? Lots of toxins and stuff that are really bad for us actually work in this way, in actually making sure that our active sites don't actually fit their substrates, and that's why they're very dangerous. Alright, so if the inhibitors stop a reaction, a coenzyme or a cofactor, alright, make reactions happen more, okay? A coenzyme, by the way, just means it's an organic cofactor. So organic molecules which aren't proteins actually assist in the completion of enzymes-dependent reactions by binding within that active site of the protein. So they fit inside of that active site in a way that actually makes the substrate a little bit better fit into that active site. It makes the reaction happen even better, okay? So in this process, the molecules themselves, the coenzymes, are broken down, and so they have to actually be replenished all the time. The best example of these coenzymes are the group of molecules that are known as ATP, NAD+, or its charged form NADH, and NADP+, or its charged form NADPH. They're loaded forms, all right? And they're three familiar organic molecules that all have roles within the two energy transformation reactions in the cell: cellular respiration and photosynthesis. Okay, these enzymes actually carry energy in the form of protons and electrons within their bonds to the active site of specific enzymes within what we call those loaded forms. Okay, so ATP, NADH, and NADPH are the loaded forms. Once these bonds are actually broken, they take on what we call their unloaded forms. So ADP, NAD+, and NADP+ are their unloaded forms, and they separate the enzymes and are ready to replenish their energetic bonds later on. Let's look at ATP, the most famous of these coenzymes, and how it actually works. So ATP is here, so it's an adenosine linked to a ribose sugar, all right? And there's heaps and heaps of energies in between these phosphate groups of ATP, all right? So if we break these bonds, we actually get lots of energy. Where do these bonds get broken is inside what we call hexokinase, which is a cool little enzyme, all right? And so, for example, when we have cellular respiration happening, we take a glucose molecule and we put it inside of hexokinase. So we take it in to hexokinase together with this ATP molecule, all right? And inside of hexokinase, ATP gets broken down into ADP, and glucose gets broken down into what we call glucose 6-phosphate, which I don't care for you knowing all of that information, but the important part here is knowing that ATP acted as the coenzyme for the breakdown of glucose, okay?